Last time we left off with looking at the tabular view of the protein family sorter and manipulating the results by using words or protein family IDs, and also looking at the different ways you can filter down to different protein families. We also demonstrated how you could click on a single row and generate a multiple sequence alignment. But now I want to leave the tabular view, and let's click up here on the heat map view. Okay, let's talk a little bit about what these colors mean. So these sliders here, I can pull it open, and I can pull it open here, and then you can see more distinctly what the genomes are down this side, and the protein families on the x-axis. So when a particular cell is black, and notice when I mouse over that cell, it gives me the information about the genome, the protein families, and how many proteins this particular genome has in this family. So when it's black, it means that this gene has not been annotated in this protein family or in this genome. When it's yellow, it means that this particular genome has one protein in this protein family. When it's this mustardy color or light orange color, it means that this particular genome has two proteins in this particular family. And when it's this bright orange, it means that it has three or more. Note in the little pop-up window, this particular genome, which is Brucella-Boertis strain 9-941, has 6 proteins that belong to this protein family that is a mobile element protein. So that's what the colors mean. Let's stretch down and stretch over so we can see all the real estate here. And I wanted to talk about some of the functionality of this. You can scroll down and you can see different patterns within the protein families. In the next video, we're going to talk about manipulating this more, but this one's more about talking about these functions. I can flip the axis if I like. I personally like this one because I can, it has more real estate. I can grab particular genomes and move them around to where I might want them to be. So see, Kudo is up here now. So this is a great thing if you've done a phylogenetic tree and there's an area that you want to show next to that. You could arrange the genomes depending the way they are arranged in the tree and then make a figure of that. That would be pretty sweet. You can use search for things. So I could look, earlier we looked for flagella. And so look, I've got it here, and instead of showing me, I'm just having it highlight where the protein families are that have that in the name. So that's showing it that way. And you can see here's a cluster of protein families here that have flagella in the name. Or what we've used before is this keyword function. Let me clear the filter here. I can go in and I can still use the filter here. Like, remember when we got all the protein families, the unique identifiers that corresponded to the O antigen genes. And then I can say I want to filter the heat map based on that. And then we see only those protein families. And it's pretty clear here when you see things that have a problem, that are missing genes in a particular protein family. And so that's one of the ways that you can manipulate the protein family, and you can reset it here and go back to normal. In our next video, we're going to show you how to do clustering. Thanks for listening, and thank you for using PATRIC. Bye.